All right, now let's take a look at what we call polysaccharides, which are molecules that have more than two. Usually many, could be 25, 30, 100. I mean, there's really no limit other than after a while they get so big that they just can't hold themselves together. But these are very large molecules. And so how do we make a polysaccharide? Well, let's make the simplest polysaccharide by taking an alpha glucose. Well, before we do that, let's just write what we mean. This is many monosaccharides joined together. Okay, so let's draw a picture. Whoops. Let's take an alpha glucose. Thin line. Alpha glucose. I'm going to have to draw this a little bit smaller in order to make this work, so hopefully you'll be able to see it. Okay, so let's make a little alpha glucose right here with our little O in it. And this will be the one on the end. So we'll put all our links here. CH2OH up on top. HO. OH. OH. And we're going to draw this linked to another glucose. So I'm going to put my link in here. And we already know how this link happens, so we don't need to go explain that part again. Put the O in there and back. Okay, and then we'll draw this and another link. CH2OH. Oops, I forgot the H on this side. I just noticed that. Let's fix that. Put that there. OH here. OH here. And we'll draw one more. And you can see how this could go on forever. So this polymer can have hundreds of these things stuck together. So if you had glucose in your blood and you were absorbing it in your cells, you're not going to use it all at once, right? You're going to need to sort of hang on to it for a while and store it. But if it just floats around by itself, remember that your cells are a huge soup of molecules. So if glucose is in that soup, it's bumping and jostling with thousands of other molecules all the time. And the chances are good that it's going to do some reacting and do stuff that you don't really want it to do. You're trying to save it for a very special reaction that we're going to learn about later. I'll put ... right here to show that this goes on forever. And actually, we could also do that on the other side. We could put this other side could let me fix the other side. You've already drawn that. Sorry about that. But we can just put we can put another thing here, O, and that's got a ... as well, right? That continues to the left and it continues to the right. There's really, I mean, there's going to be an end somewhere, but this goes on and on and on. So what it is it's a long chain of many molecules stuck together. So what I was saying before, I forgot now. What was I talking about? It's irrelevant, I guess. If these molecules are in your blood, the glucose molecule, you kind of want to pack it up and keep it safe from being attacked by other molecules until you were ready to use it. And so by sticking it together like this, in a long chain, you can pack it all up into a big molecule that's not so easily reacted, right? So this is how we store glucose. Plants store their glucose like this. This is called amylose. And although it still ends in O's, it's not a simple sugar or a disaccharide, it's a polysaccharide. A polysaccharide. And sometimes a polysaccharide is called a starch. So the word starch is a sort of a word we use for these gigantic polysaccharide molecules. And of course, we have starch in our diet. There's cornstarch in your cupboard at home, and you use it and you add it to things. And you know why what do you add cornstarch to, if any of you like to cook? To make it get thicker. You add it to gravy. Gravies often are made by adding cornstarch. You can add it to mac and cheese too. Does it make it thicker? Yeah. And that's because these long chains form these huge long strings, right? So compared to a glucose molecule, to scale, a glucose molecule is like an ant, and this is like an elephant, right? And so when you put these into your mixture, these long chains wrap all through the mixture, and they form like a web or a complicated sort of, imagine like a big ball of string, like a big knot. And it makes the whole solution less liquidy and more thick. So starches are often used for that purpose when we cook, but they're primarily used for storing glucose to keep it sort of packaged up so it can be ready when we need it. Of course, that means that afterwards, when we want it, we're going to have to use energy to break this back apart, right? So our cells can go back, and as our cells build stuff like this, plant cells in this case, they can then go back and reverse those reactions and chop this back up into its individual glucoses for energy. So it's like a storage molecule. So the thing to notice about amylose is that it is a polymer of alpha glucose. So let's write that underneath. It's a polymer of alpha glucose only. And so it contains only alpha linkages. And if you look back at the picture, you can see that the linkages we drew were the alpha linkages, right? Same side holding hands. So that's amylose, plant starch. And it typically forms very long chains. Okay, animals do something slightly different. Animals will also put together, now this picture is going to be a little bit crazy, so animals will also make a long line of alpha glucoses stuck together, but they will also make attachments at a different spot because the animals have a different enzyme which allows them to make different bonds at different places. So if you look at the picture, what we can do is, this is going to take probably a large area of page because we're going to have to draw a little bit here, come down, and there. So bear with me, make sure you have a big spot to draw this. If we draw our alpha glucose here, and again, I'm drawing kind of small. I'm just going to put the O in the same color at the same time. So this would be attached here to another one on that side. And here is my alpha glucose, CH2OH. And then we have our OH in the middle here, our OH down here. And then we have another linkage here, another alpha glucose, like this. And then what I'm going to do is I'm going to do something different on this end. So okay, this O that attaches here, instead of attaching to another alpha glucose on the left side, these particular animal cells have an enzyme that can cause it to react with the OH up here instead. So it's the same thing. It's two OH groups interacting and are forming, it's a dehydration reaction, but it's just a different spot where it's happening. Because remember, glucose has lots of OHs, right? But it requires a different enzyme to make the right shapes to make this happen. So what happens is this O, I'll keep drawing it in green, bonds to that OH, and so it bonds to the CH2 at the top of another molecule. And so what we have here is the CH2 part of another molecule of glucose right here. And that is fix this. It keeps moving on me. My microphone is not staying where it should. Not sure why. Okay, so now let's put these black lines in here. So you can see that it's attached in a different spot. It's a different kind of linkage. All right, we're not going to get into the details of it. We're just going to show that it causes this branching. But what it does is it connects to another sort of chain. And this chain might go this way, and then there might be another linkage here. All right, so CH2OH up there. CH2OH up there. OH is all around. We'll put this one as a dot, dot, dot. We'll put this one on the other end as a dot, dot, dot. And there you go. So it's just a whole bunch of these. So instead of being a single long chain like amylose in the plants, it's many chains linked together. I'm going to pause and let you copy that. Okay, so I notice we have to add an OH on here and over here. We missed those before. And so this gives us this branching sort of starch, and we call this glycogen. Glycogen. It's an animal form of starch. It's usually made in animal cells because animals have different enzymes that allow them to make different kinds of linkages. So to compare, amylose, in terms of its shape, is basically a long, windy line, right? And so amylose molecules would look like that. But glycogen molecules, if we just look at their shape, they are long, windy lines which also can bond to other long, windy lines, and they create more of a complicated sort of pattern like this. So it's a bit more of a goopy, thicker, more interacting molecule. All right. There is one other type of way that we could join. Well, there's many types of ways, but one other example I want to show you, and that is what happens if we look at a beta glucose situation. So let's take a beta glucose. Here we go. And so in the beta glucose, it's going to be a beta glucose with an alpha glucose alternating. Beta alpha, beta alpha, beta alpha. So what happens is you get this cross linkage happening. And when this happens, it creates a slightly different shaped molecule. Beta linkages are being formed. As we saw with lactose, it's kind of the same thing, but it's just a whole bunch all in a row. So we get these beta linkage, beta linkage, beta linkages. And this goes on this way, and it would go on this way, whatever. So it's the same idea as amylose, but because it's an alpha beta glucose alternating, and we get beta linkages, this is a different kind of starch. I guess I should put the OHs in. I was kind of going fast there. This starch is called cellulose. And of course, cellulose is found where? In cells. What part of cells and what kind of cells have cellulose? Plant cells in the cell wall. Animal cells don't have a cell wall. Cellulose is made by plants in the cell wall, and what it does is it gives rigidity. These complex string-like structures are almost like trusses or beams that help strengthen and give rigidity to plant cells, allowing them to build tall tree trunks and, pretty strong structures. The thing is, though, because it's made of beta linkages, you need a different enzyme to break them. It turns out humans don't have this enzyme. And so humans cannot chop this one up into glucose molecules. We can chop up amylose, because it's an alpha linkage. And we can chop up glycogen, because it's an alpha linkage. But we can't chop up this one. That leads me to the question you asked before about whether or not the second linkage in glycogen is an alpha linkage. I'm going to say yes, because we can chop it up. So we have the enzyme. I'm going to guess on that. Anyway, we can't break this down. So when we eat it, it stays stuck together in our system. It goes right through our stomach and through our intestines, and it goes right through our colon and comes right out. But guess what? If you have these big long chains of this fibrous molecule in your intestines, they help push things through. And so when we talk about eating fiber, that's what we're talking about, is this cellulose starch molecule that we can't digest. But it's still good for us because it serves a different purpose. It allows our intestines to move the contents more easily through, and it keeps everything flowing. So we don't get nutrient value from it, but we get a second sort of option, which is, this helping our intestines go. So when we talk about fiber in a diet, what we're talking about is this cellulose starch, which we cannot break down because we lack the enzyme to break down the beta linkage. But many animals can. Animals that can subsist entirely on eating grass, they have other options. First of all, they have more complex stomachs to help digest things more thoroughly, but they may also contain different enzymes that humans lack that allow them to break down cellulose. Bacteria can do it. If you eat a lot of fiber, just like the lactose intolerance, if you eat a lot of fiber, this stuff goes into your intestines. And the bacteria there will happily break this all up, and in the process use the glucose that's left over to burn and produce gases. And so if you eat a lot of beans and fibrous stuff, you tend to get a little more gassy. But that's the trade-off for pushing through your bowels. So these are the three polysaccharides that we're going to talk about. Amylose, glycogen, and starch. They are basically polymers of glucose. Now, of course, you can have polymers of glucose and fructose and all the others, and there's many different starches and many different options, but we just want an example to help us understand the concept. So these are the only three that we're going to study in any real depth. Polymers of glucose. And because they are polymers of glucose, they are all called polysaccharides. So there's the disaccharides, which contain two, and there's the polysaccharides that form longer chains. Right? All right, so that's enough for today. Like you